This conference will now be recorded.
Okay, so we're going to go through here relatively quickly, kind of a review of some basic properties of cells, molecules, how these things are going to kind of be foundational. Uh, my expectation is that you guys know most of this stuff, right? Um, this had certainly better not be your first rodeo with cell biology. Um, if not in high school, certainly here in college, we've covered this stuff. So while we are going to review it, excuse me, while we are going to review it, it is going to be expected that you guys know most of the stuff already. All right. So what we're going to do as we go through here, right, we're going to kind of raise our way through the four really important cell-related issues here for us in this course, which is to say the formation of barriers, right, cell membranes, the formation of barriers as epithelia, the functional proteins, the enzymes, and of course, how do cells talk to each other, cell signaling. Now, as again, I expect you guys to know, the cell membrane, I'm old, I call it a cell membrane. I know that you guys call it plasma membranes nowadays, but I'm old, so you're just going to bear with me. Is a, it's a barrier, right? The idea here is that it keeps the outside out, the inside in. And it does so, right, by being made up of these special fats called phospholipids. And this is actually pretty important for us, right, because the lipid part of it, the middle chunk of our little sandwich there, is hydrophobic. It supposedly keeps water that's outside, and things that are aqueous outside, outside, things that are aqueous inside, inside, right? Um, in practice, it actually does not serve as a barrier to water at all. Water molecules will force their way through this cell membrane if they want to. But it does serve as a barrier to things that are dissolved in that water, things that are going to be hydrophilic. So your amino acids, your carbohydrates, things like that are effectively blocked by this barrier. The phosphate part of this, of course, becomes important because the outside and the inside of this membrane are both in contact with aqueous solutions, and they have to be able to interact with it. If it was fatty, they would not interact with it. The phosphate group on the outside and the inside of this barrier serves as the interaction point, the hydrophilic part of this, allowing the cell to exist, both aqueous inside and aqueous outside. Now, of course, embedded within this plasma membrane, right, we're going to have proteins, either as structural proteins, as channel proteins, as any number of different tasks that they're going to perform. We have cholesterol scattered throughout, although not randomly, tends to be clumped together. Cholesterol there, of course, serving to create relatively stable rafts within the plasma membrane so that we can have attachment of things and stuff like that. And of course, on the outside, we have the candy coating, right? The carbohydrate layer that serves as name tags and a number of other purposes with that. So it's a pretty important little thing here for us. Again, if we think about the structure of these phospholipids, right, it's just a triglyceride, right? A glycerol molecule and our fatty acids, only instead of having 3 fatty acids and thus being a triglyceride, we replace one of those with this phosphate group here, right? Interestingly enough, with choline as the attachment to the phosphate group. And thus we have a polar molecule, right? We have one side that is hydrophilic, right? It will interact with the watery solutions on either side, and the middle sandwich here that is hydrophobic, that serves as that barrier to diffusion of things dissolved in that aqueous solution. And of course, the other part of this, right, is that because this is fatty, anything that is lipid soluble, this barrier is invisible to it. So steroid hormones, things like that, will pass, just blaze on through there like it's not even there. Right. And again, this arrangement here is relatively important, right? Because again, we have the need to be interactive with water on the outside and on the inside. And so we end up actually with this kind of automatic arrangement of these molecules with these hydrophobic tails in the middle and the hydrophilic heads on the margins. Now, one of the things, if we go back to this little cartoon here, one of the things that we should talk about here is that these phospholipids, like any lipid, can come in different forms, right? And by that I mean saturated versus unsaturated. A saturated fatty acid is one where we have a full complement of hydrogen atoms on the fatty acid, right? So if we look over here on this one right here, if we look, our fatty acid chain is entirely CH2, 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 all the way down to the one at the very end where, of course, we have CH3. Single bonds between every carbon, right? An unsaturated fatty acid, on the other hand, right, is going to be one with at least one, and sometimes more, double bond, right? It is not saturated with hydrogens because there are places where we could put more in, but we have a double bond instead, okay? And the reason that we care about these, right, is that these two different forms of hydrocarbon, right, have different physical properties. They are more or less flexible or bendy or malleable, right? And so what we find is that in certain cases, so here what we're looking at right now is the phospholipids that we find in various species of trout, or various individuals of trout even for that matter, or other fish that live at different sorts of temperatures, okay? And the constraint for us here is that we want a cell to be, an animal cell, to be a little flexible, right? It's not a plant cell, it doesn't have a wall, we want it to be a little flexible. We need it to be a little flexible. That's how animal cells work, right? We don't want it to be too flexible, right? We don't have too much give because then it's not, functional for us, but we want it to have a little bit of give. And so what we're going to do is we're going to lean on the property of saturated versus unsaturated hydrocarbons, right, phospholipids, to give us the amount of flexibility that we need. The other variable here is temperature, okay? And what we find is that the warmer the temperature, the more flexible any given cell membrane is going to be, okay? So what we see when we look at these different species of fish at different typical habitats is that fish in warmer waters, so our tropical fish down here, for example, their cells maintain flexibility through temperature control. It's warm, okay? And so they don't need to have, excuse me, they can have almost all of their phospholipids fully saturated, right? A fully saturated phospholipid is more rigid, is stiffer, okay? An unsaturated phospholipid is more givey, more bendy, more flexible. And so fish that live in cold waters, right, because the cold water wants to make their cells more rigid, they compensate for this, right, by having more unsaturated fatty acids in their cell membranes. Membrane proteins, right, are going to be embedded within this cell membrane, obviously, either fully or partially. And they're going to do basically one of 5 different jobs for us, right? The first 2 here, I think, are reasonably obvious, right? If we have this membrane that prevents most things from getting across it, well, we need to have some way of getting things across it, right? Your cell needs things. Your cell produces things it needs to get rid of, right? And we have to have a means of crossing that barrier somehow. And so we use proteins for that. Now, these proteins can either be channel proteins, right, which is just a protein with a hole in it, and anything that fits through the hole can diffuse into or out of the cell, depending on the concentration gradient. Or it can be a transporter, right, where it has a specific relationship with one or maybe more different molecules that it will move across the membrane, but only those molecules, right? Typically, this is active, right, in the sense that it is non-diffusive. It is requiring energy to make happen. Although, right, there are also cases where we have things like facilitated diffusion, where we use a concentration gradient, or we use some other form of energy to move things across that barrier. So we oftentimes with facilitated diffusion, right, what we do is we actively move one thing across the barrier, and then we use the gradient we create by moving it to bring something else with it. So for example, in your intestine, one of the things that we do there commonly is pump sodium out, right, of the cell. But as sodium diffuses back in, we use that energy of the diffusion to bring glucose out of the digestive chyme and into the cell, right? That's facilitated diffusion. We're not spending energy specifically to move glucose, but we're using kind of the energy created by another process to do it. So those are our two kind of movement sets of proteins: transporter, carrier proteins, or channel proteins. We also use proteins embedded in the cell membrane as enzymes, right? Our little molecular chaperones, right? Are ones that are going to make chemical reactions easier to accomplish, right? Reduce the amount of energy required to make them happen. We have proteins that are going to be receptors, right? They're going to bind to a cell signaling molecule, to a ligand is the technical term for this, right? And it's going to affect some kind of change inside the cell so that the ligand doesn't have to enter the cell. And then sometimes they're going to be structural. They're going to be cell adhesion molecules. They're going to be internal structure for cytoskeletal elements. They're going to be anything that's going to give the cell itself some shape, some structure, some rigidity, right? Some form. Now with proteins, the important thing here, of course, to remember is that shape defines the function of a protein, right? Proteins work by interacting with other molecules in kind of a lock and key mechanism, okay? And if they don't have the right shape, the keyhole doesn't work, all right? And so they can't function in the way that they need to do. So because of that sort of dramatic or intense reliance on a particular shape, we actually find that they assemble themselves in a series of different kinds of levels of increasing complexity, right? So we talk about primary structure, right, which is just having the right amino acids in the right order, okay? And from there, what we find is that this primary structure, different amino acids, because they have different R groups, are going to have different attractant forces to other amino acids. We're going to start seeing that certain amino acids will form hydrogen bonds with other ones. And depending on where those amino acids are in that chain, right, this long string of amino acids is going to form a different secondary structure, a different shape, right? So sometimes what we find is that these tend to coil up like a spring, right? So we get alpha helices formed from this. Sometimes they tend to fold up like sheets, okay? So we get beta sheets or pleated sheets. And within a single protein, we're going to find that these two forms, alpha helices and beta sheets, alternate, right? Some sections will be in helical form, some sections will be in sheet form. It's not one or the other within a protein, it's both of them interspersed with one another. And then beyond that, right, we're going to again continue to take that secondary structure and continue to fold it, right? We're going to continue to have bonds forming. That's going to take those coils and sheets and bend them further to where we get to tertiary structure, right? So the big three-dimensional structure. And sometimes we can stop there, right? Sometimes this is enough, right? So myoglobin, as our example here, or pyruvate kinase, is enough here to be functional, right? We've formed the keyhole, right? We're functional, we're good to go. Sometimes, though, we're going to be a larger molecule than that. And so what we end up with, something like hemoglobin, is taking 4, in that particular case, it can be any number, different tertiary structures and binding those together and sticking those together, right? So here in our quaternary structure diagram, we have a purple and a lavender and a salmon and a red protein, right? Each of which has its own tertiary structure. We stick those together to form a quaternary structure. So sometimes that happens, sometimes it doesn't, right? And again, we can draw these out in all kinds of different ways. You'll see them drawn out in all kinds of different ways. They're all kind of different, trying to represent the same thing. Okay. And again, I'm not so worried about this. Let's keep that. Okay. So cells by themselves are, so I've used this analogy before. You might have heard me use this before. There's kind of this weird thing that happens with people, right? Where a person by themselves can usually be relied on to make reasonably good decisions, right? A person by themselves probably not going to walk down the street and say, man, I should really throw a brick through the window of that Taco Bell, take all their money, right? But you get a bunch of people in a group together, and all of a sudden that kind of shit happens, right? I went to Michigan State for my undergrad, and around this time, following a series of NCAA tournament losses, there were occasionally riots in the streets. Again, people that were going to college, right, were ostensibly smart people, but they got in a group and they did some dumb stuff, right? With cells, it's the exact opposite. Okay, cells by themselves usually can't do much, right? They're not very smart. But you start sticking a whole bunch of them together, right? Now you can do some pretty magical things. Okay, so we oftentimes don't find cells operating by themselves. We usually find cells stuck together with other cells that are like them in tissues, right? Now, specifically here for this particular chunk of the course, we talk about cells here acting as barriers, right? In the same way that cell membranes act as barriers, but in this case we call them epithelia. With epithelia, we find that, like with all structures, right, form and function are pretty tightly linked, right? So, for example, right off the bat, one of the things that's true about epithelia is that they're avascular. They have no blood supply of their own, right? So they're always going to be attached to some connective tissue, right? And that connective tissue is going to be what supplies the nutrients and takes away the waste products for the cells in the epithelium. So what we're going to see with all epithelia, right, is that we're going to have a basal layer, right, that sits right on top of the connective tissue and is essentially glued to it or connected to it by something called a basement membrane, right? On the other hand, because epithelia are barriers, right, they're coverings and linings, okay, there's also always going to be one side that is not attached to anything, a free margin, right, or an apical margin, okay. In some cases, like we see here, our epithelia are what we call simple, which is to say there is a single layer of cells. Every cell touches that basement membrane, right? And these can be, in this case, we've got simple cuboid. Here we've got simple columnar. We can also, if the cells are flat and sort of scale-like, we can have simple squamous, right? But we're going to define them based upon the number of layers of cells and the shape of those cells at the apical layer. Okay. So here's another example, right? Now, instead of forming kind of a outside covering like you might see there, here now we're forming a tubule, right? Or a follicle. All that has to happen, right, is we have one edge that's not connected to anything, that's free, right? And one edge that is on connected to some connective tissue here at a basement membrane. If we have, kind of see if I can find a good example of this. I don't have a good example here. If there were, for example, another layer of cells on top of this, right, that would be a stratified epithelium rather than a simple epithelium. In terms of forming tissues from groups of cells, right, we have what are called cell junctions. Okay, cell junctions that exist to be essentially watertight, okay, are called occluding junctions. Typically, we talk about tight junctions, okay, so something like this example over here, where they are the cell membranes are in physical contact with one another at several points throughout the junction itself. Okay, so a tight junction, something like this, or a septate junction like this, they're essentially the same. For our purposes, they're essentially the same. And again, these are not particularly tough. You can pull these cells apart, right? But what they are is watertight. So if you have, say, digestive enzymes, right, digestive fluids on one side, you want to keep that inside your stomach. Okay, if it gets out of your stomach, it starts digesting your organs. And that's not really what you're shooting for, right? So we have tight junctions or septate junctions, these occluding junctions, to keep fluids on one side or the other. If we're concerned with structure, if we're concerned with cells being stretched and pulled apart, things being put in tension, okay, and then we're going to have something called a desmosome, right? And a desmosome, the actual structure of it is almost exactly like how Velcro works. We have these plaques here, these thickenings on the inside of the cell, and these little glycoproteins that stick out and kind of attach themselves. I'm doing something on my hands, of course you can't see it. But they attach themselves to each other like Velcro, and so they become very, very tough. They prevent cells from being pulled apart. So we see this sometimes together with these occluding junctions, right? So your stomach is capable of stretching, for example, right? We want to have both of those present there, okay? We also see desmosomes in your skin epithelia, for example, any place where we might have some stretch happening. And then finally, one that's not really so much a structural thing, but more of a communication kind of thing. These are gap junctions, right? And what these do is they form what's called a functional syncytium. Okay, basically it takes two cells and makes them one cell. So the cytoplasm here and the cytoplasm here is directly connected. They are in communication with one another. Okay, we have these little pores that are connecting them, that are functionally giving us a pathway directly from one to the other, built out of these proteins called connexins. And they essentially mean that whatever happens in this cell also happens in this cell essentially simultaneously, right? And so it's for rapid communication. So we see these things in like smooth muscle, in cardiac muscle, stuff like that. All right, and we've kind of already covered this notion of the apical versus the basal layers of the epithelia, so I'm not going to worry too much about that. In terms of getting stuff across a cell with a tight junction, we have a couple of different options because we do still need to move things into and out of some of these regions. So for example, in your small intestine, you need to be able to absorb nutrients into the bloodstream, right, out of the chyme that's passing through it. You want the fluid to stay where it's at, but you want specific things to be able to cross over. And so we can either move them through transcellular pathways, right? Enter them into the cell, and then pass them back out of the cell, the other side. Or we can, in some cases, have them move between the cells across these tight junctions through a paracellular pathway. Okay. Now for probably the most interesting thing of the whole unit here, of course, are enzymes. And enzymes are, again, things that I assume you have some background knowledge on. What they do is they simply make chemical reactions easier, right? They don't directly participate, right, in the sense that they don't get changed, right? So we have reactant, right, pyruvic acid and hydrogens, and a product, right, our lactic acid at the end of the day. We convert those two things. We take the hydrogens, pyruvic acid, we make lactic acid. All the enzyme, in this case lactate dehydrogenase, all the enzyme is doing is simply kind of making it easier for the hydrogens and the pyruvic acids to meet, right? Lowering the activation energy is the technical term for it, but it's more or less like having a matchmaker, right? You go to a party, you're there, you're single, you don't know anybody, you have a friend that's hosting the party, they bring you to the partner of your particular predilection and say, hey, have you met this person, right? I think you guys would get along. That's what an enzyme is doing. That's what lactate dehydrogenase is doing here. Okay. Now, that's a very simple version of this reaction. Okay. The actual reaction is a little more complicated. It involves molecule NADH2. This is going to be the donator of these hydrogens to merge with pyruvic acid here to form lactic acid. This reaction, I'm just going to warn you right off the bat, this reaction is very important. We're going to spend a reasonable amount of time working with this. And what we're seeing here is a part of the ATP respiratory pathway. The formation of NADH2 is a part of many steps along the breakdown of glucose to eventually ATP, carbon dioxide, water, and in many cases lactic acid. So it's worth taking this slide and kind of thinking a little bit about it. When we talk about chemical reactions, one of the things that we talk about here is something called reaction velocity. And you guys have had chemistry. You're familiar with reaction velocity. But basically here, the rate at which velocity increases with increasing concentration takes one of two basic forms, right? We can have hyperbolic, right? These are called kinetics, is technically what we're talking about here. And basically what hyperbolic kinetics is in the presence of a enzyme, we see that there is a relatively rapid increase in velocity with relatively small increases in substrate concentration. The enzyme here, what it's doing, right, is when there's very little concentration of our substrates, of our reactants, right, it makes it easier for them to get together. All right. Now, eventually, right, as we get closer and closer and closer to being saturated, right, as we add more and more and more and more substrate, eventually there's going to be more substrate than there is available enzyme for it, right? And so the rate of this reaction, rate of increase of speed of this reaction, right, is going to decline, right? It's not going to be as fast. And eventually we'll get to a point where we simply can't go any faster. Okay? In the absence of enzymes, right, what we typically have instead is something more like this S-shaped or sigmoid kinetics, right? Where at very low concentrations of substrate, because they have a really hard time finding each other, reaction rates are really low, right? And there's a little sweet spot in here, right, where we have enough substrate available for them to meet regularly, right? And small increases result in big increases in velocity. But again, like we see with hyperbolic kinetics, where we kind of reach a decline in rate, we see the same thing happening here. So the two big differences, the two things that enzymes are doing for us in terms of kinetics, is they're making reactions happen faster at low concentrations. And they also, you can see this here maybe, I don't know how easy it is to tell, they also reach maximum velocity faster, right? At lower concentrations, they will happen faster, and they will reach maximum velocity faster as well. So some pretty important stuff. And they do this, again, just kind of thinking about this simply, right, by reducing the amount of energy, right? We think about chemical reactions in a very abstract sense here. And all they really are is just two molecules that will react with one another, literally randomly bumping into each other, right? And the way that we increase the probability that they'll bump into one another, okay, is through increasing the energy in the system. Typically, we talk about this in terms of temperature, right? The warmer a substance is, the more the molecules bounce around. And the more they bounce around, the more likely they are to run into a willing partner, right? So typically, to make a reaction happen, right, we have to add heat. Right? We have to add energy, typically, again, in the form of heat. What an enzyme is doing, because it's forming matchmaker, right, it's performing that job, we don't have to add as much heat, right? A reaction will happen with significantly less energy input, right? That's really all it is. Now, enzymes often come in multiple different forms, okay? We can talk about isozymes as different forms of the same enzyme. And for animals that experience multiple different kinds of internal conditions, so say if you're a conformer for temperature, right, you might experience temperatures in the 20-degree range, the 25-degree range, and the 30-degree range throughout your day, right? You're probably going to have a different enzyme that works optimally at each of those temperatures. Okay. And what we're going to find is that what this means in a kind of functional sense is that the affinity of the enzyme for the reactants changes, right, under various conditions. How willing this enzyme is to bind to the reactants, right? And so if we think about various affinities, right, relatively high affinity here, so X is a very, very high affinity enzyme, Z is a relatively low one. What this simply means, right, is that reactions will happen faster, right, at lower concentrations under X than they will under Y or they will under Z. Now, sometimes we can also use these to control the rate of reaction, right? So sometimes we don't want a reaction to happen as fast as possible, right? So we might produce, in those cases, you might produce, say, enzyme Z, right? And if we need it to speed up, we get rid of enzyme Z and we make enzyme X instead, right? The way that we're going to compare these enzymes, right, is with something called, K sub M, right? K sub M here, we're thinking about basically the halfway point in terms of velocity for us, right? They're not going to be sort of the topmost, or how do I put this here? Basically the idea, it's called the half concentration constant. I guess we'll say that. And all this really ends up being, is basically a measure of how saturated, how much substrate do we have to reach half of the maximum velocity? Okay, maybe that was overly complicated. I don't know. The relationship between affinity and K sub m, right, is inverse. So the higher the affinity, the lower K sub m is. And remember, K sub m is our substrate concentration. So a high affinity enzyme requires comparatively less substrate. A low affinity enzyme requires comparatively more. So K sub m will be higher with low affinity and lower with high affinity. All right. Now, as far as how proteins actually work, whether they're enzymes or other forms of proteins, again, they require something called an active site in order to be functional. This is what all of that structure was about building. This is the keyhole, right? So here we have an enzyme called hexokinase, right, that binds reversibly with glucose. The quaternary structure of hexokinase here is such that we have a very specific catalytic vacuole, it's the technical term for this, right? This little guy right there, that is determined by the shape of the protein itself. And it, in this particular case, binds reversibly with glucose, okay, but it only fits that particular molecular shape, right? It's very specific for this particular ligand, is what this chemical that binds to a catalytic vacuole is called. Again, we've talked about this notion of there being different forms of the same enzyme, oftentimes in the same organism, right? These are called isozymes. So we've got our old friend LDH. We showed LDH up there before, lactose dehydrogenate. In humans, for example, we have multiple forms of LDH. We have LDH A4, B4, C4, right, that are present in our case, different organs. Okay. Same is true actually for rats, right? They have 3 different isozymes of the same enzyme. Again, LDH A, B, C. When we have essentially the same enzyme in different organisms, we call these homologs. Okay, so different forms of an enzyme in one animal is an isozyme. Different forms of an enzyme in different animals are homologs. And again, the idea here is that simply that we have seen that these these enzymes have evolved in the same way that all of the other structures of an animal have evolved to perform specific tasks under specific internal environmental conditions. One of the things we talked about before, kind of just touched briefly on when we were looking at reaction velocity and affinity, is the idea that we can use enzymes to regulate the rate of a chemical reaction, right? So here over on side A here, we have an example of a multiple reaction pathway, right? That's going to take us from A all the way to our final product E, okay? And everywhere along the way, right, at each of our 4 different reactions, we have an enzyme that catalyzes it. In the reaction from B to C, right, this enzyme, right, is relatively slow, right? Because we can't go from C to D until we go from B to C, right? B to C basically sets the pace for the whole reaction, right? If we go fast B to C, the whole reaction goes fast. If we go slow B to C, the whole reaction goes slow, right? We don't have to affect every step in the pathway. We can just affect one of those steps and have pretty profound rate changes. Another thing that we can do with enzymes isn't necessarily related to reaction velocity or rate of the whole chain, but we can actually take the same reactant and make different things out of it, right? We can have branching metabolic pathways as well, right? Depending on which enzyme we use, right? So here over on B, right, we go from A to B, okay? If we have enzyme 2 present, right, we react to turn B into C, and we go down B, C, D to E, this particular pathway here. If we get rid of enzyme 2 and instead produce enzyme 5, we don't go down this pathway. We don't end up with E, right? We instead go down this pathway and we end up with H instead. So, again, this is an example where we can do something relatively simple. We produce this protein or that protein, and we end up with, in some cases, radically different products from the same reaction chain. This is an example here of one of my favorite kinds of reactions. This one's a little bit hard to understand maybe. This is called allosteric regulation. Okay, and the deal with allosteric regulation is actually pretty simple. It's not as complicated as you might imagine, although it does look pretty difficult. Basically, the idea here is that we can take a enzyme, and by combining it with one ligand or another, we can change the rate of the reaction. So we don't necessarily have to change the enzyme. We can simply bind it to something different if we want to, right? So in this particular case, right, again, this is a massively important pathway for us. We spend a lot of time talking about this one. This is super important. This is the pathway of breaking glucose down to pyruvic acid. This is the first step in ATP production for most of the reactions in your body. Okay. The step between going from fructose 6-phosphate to fructose 1,6-diphosphate, right, is regulated by a enzyme called phosphofructokinase. Okay, and phosphofructokinase can go fast or it can go slow, right, depending on what ligand binds to it. So in this case, we have two different potential ligands for us. We have citrate and we have adenosine monophosphate, AMP. Citrate, right, downregulates, okay? It makes the reaction go slower, right? So if we don't have a real severe lack of ATP, we'll bind citrate, phosphofructokinase will go slow. We won't burn through glucose nearly as fast, okay? On the other hand, if we're using a lot of ATP, right, we're taking ATP and converting it to ADP, and sometimes we're converting that ADP to AMP, right? We're stuck with a lot of AMP, right? So the presence of that AMP, because we're burning off a lot of these phosphate groups, right, is going to then instead bind to phosphofructokinase, right, and cause us to increase the rate of ATP production. It's going to up-regulate rather than down-regulate, right? And so this is what allosteric modulation is. I think it's fascinating. I think it's really cool. It's just kind of using one enzyme to make things go faster or slower depending on the internal conditions of the cell. I think that's pretty neat. All right. The next kind of modulation that we've got is something called covalent modulation. And covalent modulation basically is just the making or breaking of covalent bonds between enzymes and their modulators. Two different real options on this. We've got kinases and phosphatases. Okay, kinases serve to phosphorylate things. Okay, so they add energy to a molecule to do something. A phosphatase removes a phosphate from something. So if we go to our example here on our slide, at the very top, right, we have this enzyme over here with an OH group. If we're going to take a phosphate group, if we're going to phosphorylate, we're going to add energy to it, we're going to add a phosphate group to it, right? We're going to go this direction, right? We have to use a kinase, right? A kinase phosphorylates it. On the other hand, if we want to take this phosphate group and break it off, okay, this case, we use a phosphatase. We remove the phosphate. We take the energy in that phosphate bond and we give it to somebody else. Kinases, because they tend to add energy, right, are often involved in these kind of cascade reactions, right, where a single activation, if I can find my cursor again here, we find a single activation of one cell, we take this energy and we use it to phosphorylate multiple cells, multiple enzymes, multiple proteins, whatever, okay? And those in turn also activate multiple proteins, right? They phosphorylate multiple molecules from then on. And so we start with simply one activity, but we end up with this big cascade of reactions, sort of an amplification is what it's called. Okay. Now, one thing that we'll spend some time with in a little bit is tracing the evolution of isozymes and of homologs. Okay, so here we've got kind of a little evolutionary tree, if you will, of LDH enzymes. And one of the things that is interesting to me out of this whole thing is that isozymes tend to group together. They have an evolutionary history independent of the organisms, right? And what I mean by that is if we look at, say, LDHC here, for example, all of the homologs of LDHC group together, right? They're all found in this group here. The isozymes, the homologs for LDHA, right, all group together here. It doesn't matter what the organism is, right? Here's human LDHA and rat and mouse and all this other stuff. And here's LDHC, human and rat and mouse and fox and all these things. LDHB, right? And so these things evolve not as organisms evolve, but they evolve as needs require for groups of animals. I just think that's interesting. All right, let's see, what do we want to do here? One of the things that is oftentimes pretty interesting, at least to me, is that we can often find ourselves looking at the same species with a relatively large range, but we begin to, because they live in different environmental conditions, different isozymes are more prevalent. Okay, so here we've got killifish, this little fish. You can buy these at the pet store. They're little tiny little guys. But they have this particular species of killifish has a large range in the Atlantic coast, right? All the way from Maine down to here in Georgia, South Carolina, North Carolina. You might imagine the environmental conditions are pretty different for this range here, right? Relatively cold water up here, relatively warm water down here. And just as we saw, things like the phospholipid saturation change with temperatures, here, because we live in different ambient temperatures, we have different amounts of activation energy available to us, right? Very little over here in the cold, quite a bit down here in the warm. And so we start to see separation between which enzymes are produced in what quantities by, in this case, fish in different parts of their range. This might eventually lead to a situation where we actually have a speciation, right? Where we have sort of a northern species and a southern species, where right now we have a single species. Another fun reaction here, or fun aspect of these reactions is, of course, enzymes have to be able to change over the course of an individual's life, right? Our old friends, the acute and chronic scale changes in physiology, right? And a big part of that is that at some point or another, we either are going to have to repair damaged proteins, or we need to get rid of proteins that we no longer find useful, right? So we have a set of proteins called molecular chaperones, right? Oftentimes they're called heat shock or stress proteins. And the jobs of molecular chaperones are to repair damaged proteins. On the other hand, what we're seeing here, okay, is something called ubiquination. Okay, and basically what ubiquination is we literally stick a little thing here called ubiquitin onto a protein. Basically, just it tags a protein for removal. Right, and the more ubiquitin we stick on a protein, the more likely it is to get removed. So we see things like polyubiquination. Okay, and eventually what's going to happen here is this ubiquitin chain becomes a target for something called a proteasome. A proteasome basically is just is an enzyme, okay, that takes a protein and breaks the bonds between it, breaks the peptide bonds between the amino acids, and essentially recycles it. Okay, so we get to that point where we can remove things that are no longer necessary for us. One other little fun thing in here, just kind of a fun thing, bioluminescence. We talked about the luciferase reaction in fireflies from before. It's basically the same reaction that occurs in all bioluminescent things. For us, and if you work with Dr. Engel, I think maybe Dr. Golden does some of this stuff too. One of the things that we do to track protein expression in cells, if we're trying to do experiments with things, is we tag that particular protein with something called green fluorescent protein, okay, or GFP. We actually harvest GFP from the luciferin cells in this case, jellyfish. So just kind of a neat little tack on there. We do have some instances where bioluminescence is symbiotic. So we get a number of squids, not every squid, but a number of squids absorb bioluminescent bacteria, and they actually install them in the cells on the undersides of their body. And the reason that they do this is to produce what's called counter illumination, right? So if you're a squid and you're swimming along, right, you cast a shadow on the ground. And if your prey is living on the ground, they can learn to avoid you by simply avoiding your shadow, right? If you glow, you don't cast a shadow. You are essentially invisible to the things that are on the ground. And so it's a pretty neat reaction. And then this is just an example of a squid that does do that. Here's an example of what that looks like in practice, right? They are going to stick out these little epithelial projections to attract these bacteria called Vibrio fischeri. Okay, and the Vibrio fischeri then colonize the tissues and give them bioluminescence. Okay, so that's good for us for now. I will see you guys on Monday. We will finish wrapping up the whole cell thing and move on with our day.